Так, что мы можем...
Okay, what can we do here?
Итак,
Okay,
меня слышно, наверное, да?
I hope you can hear me. Yes, we can hear you.
Дорогие друзья,
Oh, dear friends,
для меня сегодня выпала
I
честь
think it is a great honor for me to introduce the lecture
из выдающихся мировых
of one of the most well-known world scientists who is currently researching the nature is searching for new ways how to diagnose, how to treat very difficult and severe brain disease related to the neurodegenerative process like AD or PT, and these diseases brings a lot of suffer to the millions of people in the world. Professor Hardy made also significant discoveries in this area.
Я могу продолжать?
I hope I can go on.
У меня какой-то очень громкий звук в...
Unfortunately, I had quite a very off noise in my computer.
Профессор Харди обращение к директору и организаторам. Меня перебил...
Unfortunately, there is another voice which is currently interrupting me because I get a kind of loop of my own voice.
Профессор Харди,
got a PhD at Imperial College, and he has been initiating the AD research since 1980s at this university. These were the first pioneering researches.
Это были пионерские исследования, которые позволили... Я прошу прощения, я все время слышу громкий...
I get my voice back.
Да, я просто не могу говорить.
Unfortunately, I cannot keep talking.
Можно выключить этот канал, который резонирует? Мое выступление поэтому будет очень коротким, чтобы успеть сказать. Профессор Харди сделал целую серию выдающихся открытий в этой области. Например, он выяснил, что именно мутации в гене предшественники белка амилоида вызывают отложение бляшек и накопление отложений в нервной системе. И ряд других генетических механизмов, которые были открыты вначале в области болезни Альцгеймера, потом болезни Паркинсона и других нейродегенеративных заболеваний. Профессор Харди сейчас возглавляет кафедру и отдел в университетском колледже Лондона. Но он прошел большую карьеру, работав в университете Южной Флориде в США, в клинике Мэйо, в Национальном институте здоровья в Бетезде и вернулся в конечном счете в Лондон, где продолжает сейчас работать. Он получил большое количество премий за эти. В частности, одна из премий 2015 года – это так называемый Breakthrough Prize, премия за прорыв в области медицины, премия, которая была учреждена Сергеем Брином, Марком Цукербергом, Юрием Миллером за выдающие исследования в области медицины.
Discoveries in the healthcare or medical area.
Konstantin, perhaps I should start?
Dear John, welcome to our festival, and thank you very much for coming and giving this talk. We are looking forward.
Thank you very much, Konstantin, very much. Thank you very much indeed. So thanks for the honor of inviting me. What I'm going to do is talk about the most common neurodegenerative diseases which affect us. These are all diseases of aging, and although it's difficult to believe in these COVID times, they are a major, perhaps the major health problem that we in developed countries face with an aging population. These are the diseases I'm going to talk about. I'm going to show you the pathology first. Here you have an Alzheimer brain. This is a ventricle here. It's far larger than it should be. There's been a lot of cell loss here. And this is a cross section cut through here. And here is the hippocampus. This is the area of the brain where you make new memories. And this should be much fatter than this, much bigger. And this is the histopathology of the disease. This is a plaque. It's about 1/10 of a millimeter across. And it's made up of a protein called amyloid. These are blood vessels, and these too have got amyloid around them. And then these are nerve cells from the cortex, and inside them they have these deposits called tangles, which are made up of the tau protein. So this is the pathology of the disease. And of course, what we don't know when we look at the pathology is what order.
Дмитрий, а вы будете переводить?
Да.
У нас как бы идет уже эфир, но перевода нет почему-то от вас.
What we don't know is the order in which that pathology occurred. This is the pathology of Parkinson's disease. This is the top of the spinal cord. Here is the substantia nigra in a normal individual. And this is the substantia nigra in a Parkinson individual. And you can see you've lost this color here. And when you look in the microscope, what you see is these, and these are called Lewy bodies, and these are made up of the protein synuclein. This is a disease called progressive supranuclear palsy. This too shows neurodegeneration. This part of the brain should be larger here. And when we look here, inside neurons here, we see tangles of a protein called tau deposited. So in all three of these diseases, we see protein deposited in the disease. In Alzheimer's disease, it's amyloid and tau. In Parkinson's disease, it's synuclein. And in this disease, progressive supranuclear palsy, it is just the tau protein. Now, my group has been studying the genetics of these diseases. Most cases of these diseases do not occur in families. They're what we call sporadic. But in some cases, the diseases are early onset and occur in the 40s and 50s and are genetic. And my group has been trying to find the genes for the diseases. And the first thing is that all of these diseases can have one very simple cause. In Parkinson's disease, where synuclein is deposited, one cause of the disease is that you have extra copies of the synuclein gene. In Alzheimer's disease, one cause of the disease is you can have extra copies of the amyloid gene. And in these diseases with tau pathology, one cause of the disease is that you can have an extra copy of the tau gene. So in all of these diseases, what we and our colleagues have found is that one way of getting the disease is by making too much of the protein so it's deposited. And this point has been made from a different point of view by this paper from Cambridge by Michel van der Schoele. He pointed out that all of these proteins are very highly expressed in the brain and therefore close to what we might think of as their solubility threshold. Now, we found the amyloid gene causing Alzheimer's disease by studying this family. And I'm just showing this so you understand how genetics works. Here you have a grandfather with Alzheimer's disease, two sons with Alzheimer's disease. They married two sisters, and you're looking at the top half of chromosome 21. These 5 sibs here got Alzheimer's disease with an age of onset about 55, and so did this cousin. But these unaffected sibs did not get the disease, but these two individuals got these parts of the chromosome, and that puts the disease gene here close to the amyloid gene. And when we sequenced the amyloid gene, we found mutations. Now, in other families with Alzheimer's disease, there were other genes involved. And this gene was found by Peter Hislop's group. It's the presenilin gene. And here is the presenilin protein drawn out. It goes through the membrane 9 times. And when it was found, we did not know what its function was, but we now know its function is to cleave the amyloid protein. And so this told us that either mutations in amyloid itself or mutations in the protein which cleaved amyloid could cause early onset disease. Now, these rare causes of disease, which are all involved in amyloid production, did not apply to late onset Alzheimer's disease. And we and many colleagues have been looking for genes which predispose, they don't cause, they predispose to late onset disease. And this is how we do it, using what's called a genome-wide association study. Here are the chromosomes laying along here, and this is the significance level here. And we're looking at many markers across the genome to see which markers are associated with disease, and that leads us to find these genes here. Now, what do these genes do? Well, this very interesting paper was the first to show what they did. Nearly all of these genes that we found in late onset disease were either involved in the immune system, and in the context of the brain, that means microglia, or they were involved in lipid metabolism. So these two pathways, the immune system, which is microglia, or lipid metabolism, turned out to be important in late onset disease. And we started, this is where we made a contribution as well. We started to sequence Alzheimer cases and found mutations in this gene. TREM2 was a predisposing factor for Alzheimer's disease. Now, when we made this finding, which we made by gene sequencing, we were also looking to see which genes in the brain went up in response to amyloid deposition. And what we found was remarkable. So we found mutations, but we also found that TREM2 was a protein which went up enormously in its level in the brain when amyloid was depositing. So this is an amyloid responsive protein. And here we have, what we've done here is drawn a network of those proteins which respond to amyloid deposition. And right in the middle of this network is TREM2, but these are other proteins which respond to amyloid deposition. And these are also genes which are risk genes for Alzheimer's disease. And so what we've shown here is that how well you respond to amyloid deposition is important in late onset disease. So what I showed you in early onset families was amyloid production was important in Alzheimer's disease. But what we are now showing is in late onset disease, it's how well you clear amyloid, which is important. And that's our finding, but that finding has been made also in this paper from Randy Bateman, who showed that individuals with Alzheimer's disease showed worse clearance of the amyloid peptide. So this is the summary. The genes involved in early onset disease are involved in amyloid production, but the majority of genes involved in late onset disease are involved in the response to amyloid, microglial, and involved in lipid metabolism. Now, what I've told you so far is established fact. What I'm now going to say is what I think is going on. Here you have the amyloid precursor protein in the membrane, and in red, this is amyloid. And amyloid is cut from the center of the precursor protein. And I think what it does is it builds up in the membrane of the neurons. And when it builds up in the membrane of the neuron, it attracts microglia, and the microglia repair the damage. And what happens is eventually that damage repair is overwhelmed. And so the amyloid causes membrane damage. And if you're not good at repairing that membrane damage, or if you make too much amyloid, you're going to get sick. I'm now going to move on to Parkinson's disease. You'll remember that in Parkinson's disease, the protein which is deposited is synuclein. And we have now found many, many genes involved in Parkinson's disease, and they're all involved in two pathways. One is the endosome-lysosome pathway. This is autophagy control, lysosomes and endosomes. This is one pathway which is determining how lysosomes break things down in the cell. Lysosomes are the little organelles within cells which are responsible for the degradation, or they're involved in mitophagy. Mitophagy is the process by which damaged mitochondria are sent to lysosomes to be degraded. So over and over again, we see the same pathway, which is always involving lysosomes, which is important in Parkinson's disease. And again, as we've done the GWAS for Parkinson's disease, looking for risk genes, this is the same thing here with the chromosomes and all the genes. Most of the genes are either lysosome or they're involved in mitochondria. So over and over again, we see the same pathways. And that is also true of this finding, showing that people with mutations in this lysosome storage disease gene called glucocerebrosidase have an increased risk of getting Parkinson's disease. So early onset disease can be caused by too much synuclein. And late onset disease is predisposed to by lysosome problems. Well, what you can see in this here is that in fact synuclein is degraded through the lysosome, and having too much synuclein inhibits the lysosome. And so this is very similar to the story with amyloid. If you make too much synuclein, or you have a poor mechanism for breaking it down, you will get disease. So this is the summary for Parkinson's disease. Too much synuclein or weak lysosome function is the major reason for getting the disease. And then finally, moving on to the diseases with just tangles, we showed years ago that tau was the main gene when you had a Mendelian and early onset form of the disease. And as we found risk genes, tau is the major risk genes. These are the other risk genes that we have found, and these are all involved in the ubiquitin proteasome system. And consistent with what I've been saying is that tau is degraded through the ubiquitin proteasome system. So tau, if you potentiate the ubiquitin proteasome system, you can clear tau tangles from neurons. And so over and over again, we're making exactly the same suggestion. It's subtly different, but it's the same overall mechanism. In all diseases, overproduction of the protein can lead to autosomal dominant disease. In all diseases, many of the genes involved in late onset disease are involved in clearing those proteins. The clearance mechanisms are largely different. In Alzheimer's disease, amyloid is largely cleared by the microglia. In Parkinson's disease, synuclein is largely cleared by the lysosome. And in the tangle diseases, tau is largely cleared by the ubiquitin proteasome. And so what my overall suggestion for late onset disease is that as we get old and we have a lot of protein damage in our system because we're getting older, clearance mechanisms which are good enough for us when we're young are no longer good enough for us when we get old, and the proteins start to build up. And that is how the disease, these diseases are caused. What does that mean for treatment? Well, there's two clear ways, though they're not easy, of trying to find treatment. The first is to reduce the production of those proteins by, for example, antisense therapies, reducing the amount of the gene which is translated, or we can potentiate the clearance mechanisms in some ways. So it's pointing to either reducing production or improving clearance pathways in the disease process. Thank you very much. Of course, I'm happy to take questions. I'll try and... I'm happy to take questions or comments as questions, of course, if there is time, of course.
Dear John, thank you very much. I do not see any chat questions, but may I ask a question of my own, John? Is there any specificity in the degradation?
Is there any specificity in the degradation pathways? And I think
I see many. I hear many voices and cannot actually speak. But my question was how specific this degradation to the nervous system, and is it possible to see the parallel in the peripheral tissues in the cells of the?
There are protein deposition diseases of the periphery. For example, you can get amyloid deposition in the heart, causing heart problems, or in the pancreas, which is one of the causes of diabetes. So there are those problems that occur, and I don't know to what extent the mechanisms are similar. Of course, in most tissues, not all, but in most tissues, you can deal with losing a tiny proportion of cells every year, for example, in your liver, because you grow new cells. Whereas in the brain, of course, to a large extent, you don't grow new cells. And so that means that a slow loss of nerve cells is catastrophic, but a slow loss of liver cells can be repaired. So it's different tissues can respond in different ways.
Thank you, John.
My pleasure.
More questions from the audience.
We don't have questions.
Yes, I don't see any questions. John?
My best wishes to all of you. My best wishes to all of you.
Yeah.
Дон, можешь проговорить вопрос? Ой.
The first question is how to reduce the protein production.
So I've been very excited by antisense therapies these last 3 years, where you you inject an antisense. Well, it's like an antisense RNA molecule into the CSF, and that reduces protein production. That is being now used in some forms of motor neuron disease, and that might be a very good way, more generally, of reducing protein production.
Thank you so much. And second question.
The second question we have is, what methods do you know to reduce amyloid production?
Well, that's the same question. Using antisense. There are the other thing which people have tried so far unsuccessfully is to get antibodies to amyloid and remove amyloid. So not reduce production, but to help, if you like, removal. There have been attempts at that which have not yet succeeded, but those attempts are continuing.
So I think now we don't have any more questions. Thank you so much.
Best wishes to all of you.
Thank you. Bye-bye.